This is TWIS. This Week in Science, episode number 1049, recorded on Wednesday, February 25th, 2026, Stabilizing the Changing Brain. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with astrocytes, alcohol, and first alerts. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer! The state of all things is fine. Just sign on the dotted line. Just watch what you get and never forget. This Week in Science is coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
I don't have anyone to say anything to right now. Hello, welcome to This Week in Science. I wish I had Blair here right now. Good science to you all. Thank you for joining me for another episode of This Week in Science. We're back to talk about a whole bunch of science stories and a lot of things that happened during the last week. But I also have an interview to start out the show with. We're going to talk about the importance of astrocytes. What are astrocytes? We have so many things to talk about. So I have stories ahead, like I said, about alcohol, but it's chimpanzees and alcohol, not mine. Additionally, we're going to be talking about first alerts from the Vera Rubin Space Telescope. But right now, we're going to jump into the show, and we want to remind you that subscribing to the TWIS podcast on your favorite podcast platform is a really easy thing to do. Look for This Week in Science. You can also find us every Wednesday evening, 8 p.m. Pacific time-ish, on YouTube, Facebook, and Twitch. Twitch? No, Twitch. Yes. If you need information, head over to twis.org. But it's time for the science right now. So I'm going to introduce our guest without any further ado. Just jump right in. Tonight we're talking with Dr. Nicola Allen, a neuroscientist and professor at the Molecular Neurobiology Laboratory at the Salk Institute. She investigates how the brain normally forms and functions and how it goes awry in diseases like autism and Alzheimer's disease. She asks how non-neuronal cells in the brain regulate neuronal function. Dr. Allen and her colleagues recently published a study establishing the stabilizing role of one cell type called astrocytes in the adult brain, which is very important. She's here tonight to talk with us about her work and its implications. Dr. Allen, thank you so much for joining me tonight.
Thank you for the invitation. Really excited to tell you about our work.
I am excited to hear about it, and I hope everyone is here as well. We've talked about brain stuff and glial cells and astrocytes on TWIS a lot of different times, but we haven't ever talked with somebody who works on it. So there's a lot.
There'll be many more.
No, you're the only one ever. That's it. As we get started, though, why astrocytes? Like, how did you go from, hey, the brain is cool and development and all this stuff that's great, but astrocytes and not neurons?
Yeah, I think it's just comes back to my time in college where I was interested in the brain, but also the immune system. So thinking about how non-neuronal cells could be impacting brain function. So that led us to astrocytes, which are a type of glial cell in the brain that basically sit where all the neurons are, surround them, have lots of important roles. And around the time I was starting my studies, it became clear that they weren't just support cells, which is kind of what people had thought before, but they may have a more instructive role in telling the neurons how to function and keeping them healthy. So that's what got me interested. This was an area that had a lot of things that we still needed to discover.
Was there anything in particular that you went, oh my gosh, this is it. This is what I need to do?
Yeah, it's actually one of those moments where I attended a course as a PhD student to learn molecular biology. So my PhD was entirely doing electrophysiology, which is a technique where you can record neuronal activity. Amazing technique, but kind of maybe kind of limited the questions you could ask. So I was lucky enough to go on a course at a place called Cold Spring Harbor in the US to learn molecular neuroscience. And one of the speakers there was someone who's actually very famous in the glial biology field, someone called Ben Barres, who was at Stanford at the time. And he just gave this talk that basically showed their data that if you look in the developing brain, everyone thought neurons knew how to form connections with each other. And this was something neurons did by themselves. But what his group had just shown was actually neurons don't know how to do this if you grow them on their own. And that if you add these astrocytes, these glial cells, now you get many, many synapses form and the neurons communicate. So for a PhD student at the time in the audience, this was just an amazing presentation, an amazing discovery about, wow, there's so much to find out here. And it's kind of changed the way I was thinking about how the brain works.
When was this? Like what years were this?
In the early 2000s.
Yeah, because I remember getting into neuroscience as a graduate student, undergraduate, for certain. The astrocytes, as you said, were just support cells. It was maybe metabolism, but I remember somebody describing them as like packing peanuts, like the styrofoam, right? Like the idea that they did anything that was really important was it wasn't considered.
Right.
So do you know how we went, or how Ben Barres and others who were working on it, how they went from that conception of these cells as just being there and not part of the signaling or the development or anything? How did we go?
Yeah, so I think a lot of the groups back then were probably ahead of their time because they would just ask these questions. And I think in the field of development, many developmental neuroscientists, or even in other tissues in the body, will say, well, it makes sense. If you think about all the different cell types that develop together, there's a reason they're all there and they signal to each other to get things working properly.
Yeah.
I think now, if we look at a lot of data that's come out, sequencing data about changes in the genome in different neurological disorders, or changes in how the cells function in different neurological disorders, what we're now seeing is that it's not just neurons that change, but often these glial cells. So the astrocytes that we work on, but also the oligodendrocytes, which are the cells that wrap the myelin, the insulation around the axons, and also the microglia, which are like our immune cells that live in the brain and also have really important roles. So I think now there's a lot of excitement around all these different glial cells and what they could be doing, both in the healthy brain and also different diseases.
So you mentioned microglia as immune cells, and when you said you were interested in the immune system, I immediately was like, why not microglia instead of astrocytes?
Actually, I worked on microglia a little bit in my PhD.
Okay.
And then dabbled in other stages of our career. And now we and others. So I think it was already a breakthrough to think about not just neurons, but how neurons interact with astrocytes or neurons interact with microglia. But now the field is moving towards, okay, let's think about how all these cells communicate and they all interact with each other, because microglia signal to astrocytes and the astrocyte to the microglia and then to the neuron. So there's a constant communication going on and they're all kind of telling each other how to behave.
Which is so interesting that it's come about over the last couple of decades from this idea that the neurons were just the thing that were doing it. So how are astrocytes involved in the signaling and function of neurons? How are they not just packing peanuts, right?
Yeah, so I guess to get back to the packing peanuts comment, I mean, part of that is just their original name. So for glia, it means glue, connective tissue. So that's how they were initially described. Even though early anatomists had actually predicted some pretty important functions of these cells.
Yeah.
And then I think as neuroscientists, because we have techniques such as electrophysiology, where we can monitor neuronal activity, for the longest time, neurons were the main cell we could actually monitor and assess what was going on. And because the glial cells don't have those like fast spikes of electrical activity, so it's not something that you can pick up. So they were doing things, but you couldn't see it when you were doing the experiments. So a lot of like development of new ways of analyzing them, I think, has helped bring them to the forefront. So that's kind of one of the reasons I think they were understudied.
Right. So from the electrophysiology perspective, what would you have tossed them out as noise? Or just they didn't have the spike?
They don't have the spike. So they have interesting things going on, but they don't have the spike. Yeah.
Right. So for people who don't know about the way that neurons spike the action potential, there's this moment where a neuron gets the threshold that it's going to be active, right? And so there's what we call the electrical activity that we call a spike, and it's very stereotyped. And how different is the astroglial activity with the slow onset? Is it more of like a combination of all of them acting together? Can you measure from them electrophysiologically, or does it have to be totally different?
So you can, and people have done that, but you kind of see small changes in the kind of the potential difference across the membrane of the cell. But you don't see like a big spike. And there's also lots of technicalities of that recording technique that actually stops you seeing some things that are happening that are interesting. So instead, what got the field kind of moving forward was development of genetically encoded dyes, calcium indicators that allow you to monitor intracellular signaling pathways. And astrocytes, like many cells in the body, use increases in calcium as a way of sending messages inside the cell and communicating.
Right.
So now we can actually monitor how the astrocyte is acting within the brain, either responding to ongoing neuronal activity or having its own intrinsic increases in calcium that are then signaling to other cells. So I think that was a big breakthrough and let people see what was happening, and also see actually in some cases this is happening at the same speed as the neuron. It's just not a way that we were looking at it, essentially. Ooh.
Yeah, so I remember going through electrophysiology and also through all the courses, and there were questions sometimes about why the calcium potential was different, why threshold was different, and people had hypotheses, but I just love the fact that the astrocytes have become part of the explanation for why these differences occur. As the astrocytes, as you're looking at them, they are helping the cells. They're part of the development of cells. You've been looking at developmental angles, which means baby brains turning into kid brains, turning into adult brains. And until adult brains happen, there's learning, there's change, there's like redefining of the structure of the brain that everybody goes, oh yes, it's going to grow and change, and it's going to be different. And then when you're an adult, the idea a long time ago was, nope, that's it, no more change. You're just on the long slide down. It's just going to break from here on out. So what do we know about the role of astrocytes in the developmental period that has led to this recent paper and where we'll start talking about this recent discovery?
Sure. So I think what the work over the past couple of decades really showed was that if we think about the developing brain, so as you just said, this is a period of the neurons really going through this intense period of forming synapses, forming connections with each other. And this can be a bit of an exuberant process during development, not necessarily specific. And then they kind of get pruned away and paired back to the correct connections into adulthood. And what we and others have shown is that astrocytes actually release really specific cues that tell the neurons how to do this. So this is kind of the study I was mentioning earlier that Ben Barris's lab had done that I was inspired by, where they'd shown neurons alone, even though they're healthy and they make everything they need to form synapses, they don't. Then if you add just things astrocytes release, so secreted proteins and lipids and small factors, putting that on the neurons is enough to make them form synapses and also have electrical activity.
Without the astrocytes.
Without the astrocytes, they don't even need to be there. It's just everything that they're pumping out into the media. So that led us and others to then try and identify what those signals were. And kind of a simple idea would be, or they're releasing one thing that tells the neuron, now it's time to make synapses, and then it kicks into place a program in the neuron to do that. That wasn't the answer. It turns out they release many different factors. I don't think we know what they all are yet. I'll get to that in a minute. But they release many different things that, for example, different signals that will tell the neuron, now make a new immature connection. But that connection won't actually be able to communicate because it doesn't contain neurotransmitter receptors. So that's these proteins called thrombospondins. What we found when I was a postdoc was there's a different class of proteins called glypicans that tell the neuron, okay, make a new immature synapse, but this one will communicate because it has these neurotransmitter receptors. So that's already two very different ways the astrocyte talks to the neuron in the young brain to start forming this circuit.
Why have a non-communicating synapse?
Yeah, so this is so these are called silent synapses. They're actually very important, particularly in the hippocampus, for learning and memory. So the idea is you have this connection there ready-made, it's built, it has everything it needs to start becoming part of the circuit and communicate, but it doesn't have the neurotransmitter receptors. So if the cell receives enough information or signals to tell it, you want to increase your activity or connections, it can quickly bring in the receptors and become active.
Okay, so it's like it's there.
Yeah.
It's like pre-baked bread.
Exactly.
Yeah. Ready to go.
Yeah.
Okay, so those are there. They're not communicating. They have different proteins that hold them in place and allow them to be structurally solvent. And yet they're not communicating. And then the glyco. No,
I say glycan. You may say glypican.
Glypican. So these different synapses are the ones that get in there and they start sending signals,
firing,
which fire and create the spike, the action potentials.
But these are the sort of connection that you only see I'm generalizing here. In many brain regions, you would only see in the young brain. And if they're selected to be kept as part of the network, then you bring a different type of neurotransmitter receptor in.
Right.
And the astrocytes, again, can control that by releasing a different set of proteins.
And so the astrocytes figure this out. And the astrocytes go, you're permanent now.
We don't quite know how they have that level of specificity or whether it's the neuron making these decisions and the astrocyte provides the environment to allow them to do this. What we do know is as the brain matures, these signals that make these immature synapses in the astrocytes get downregulated, and then the signals that mature the connections get upregulated. And that isn't something the cells just do. They do it in response to changes in neuronal activity.
Yep.
So we think of it as this constant feedback between the two cell types to get things working.
To each other constantly. It's not just one to the other, one-way communication. It's actually a dialogue. So when we talk about the standard, the phrase in neuroscience where use it or lose it, it's not just the neurons, it's all of it together. Yeah. So moving from the astrocytes and the development of these synapses and the way that they work together, you ended up coming to a study where we were looking at plasticity, right? We're looking at new synapses. We're looking at the ability to create these new connections between neurons. And you found a way to look at stability and whether or not astrocytes were involved in stability. So why is stability versus plasticity important in the brain? And then what brought you to this particular question?
Yeah, so to start off with why it can be important, again, this depends on the brain region you're looking at. But if you're thinking about areas of the brain that once you've been through development, for example, you've wired up your visual system, you've wired up your motor system so that when you want to move, everything happens appropriately. Once you've been through that really extensive process of remodeling, you kind of want to lock it in, like in a broad sense. So you may have some small
changes. How to ride a bike, you want to remember.
Yeah, but you want to kind of get that circuit generally locked in. So we think in certain brain regions where you don't want ongoing relearning, then it's important to lock in the connections once they've been formed. And the reason we thought about astrocytes in this process, firstly, was because we already knew they had these really important roles in development that change across development from early life into adolescence. So you already know they change depending on context.
Mm-hmm.
And also it was really inspired by many, many other studies that had been done where researchers in this field of plasticity had shown that in an adult brain, there are actually quite a few different ways they already knew about that you could reopen windows of plasticity. And a lot of them involved changing the extracellular environment of the neurons. So changing what's around the cells. So that kind of gave us a hint that it's not necessarily the neurons in your brain that have lost the ability to remodel, but they're being blocked by things in their environment.
I think that's fascinating that there are blockers to change, that you have to maintain stability because otherwise your brain will change.
Yeah.
Right. So
Yeah.
is that am I getting it right? That basically our brain physiology, the way the cells work together, that astrocytes have to lock things in or allow things to change, and they have programs. I'm going to use that term lightly, to allow that to happen for different reasons.
Yeah. I think it also gives us lots of ideas and insights of how you could change brain plasticity.
Do you think, like, there's so much talking about brain plasticity in current media and self-help things? How much do you think is we can hack our brain and we can, how much do you think is hype versus, the plasticity it's great. And this is, yes, of course, our adult brains are plastic. Use it.
I think they're more plastic. It's not like this black and white kind of plastic in youth, not plastic in adulthood. I think it's all kind of levels of things, and we're on gradients. I think from our point of view, we're interested in thinking of situations where perhaps you would want to increase plasticity for brain repair.
Yeah.
So for example, after a brain injury or a stroke, if you have damage to the neurons, but the neurons surrounding that area have survived, we know from lots of work that there are things that then inhibit those surviving neurons from repairing the connections. So we're interested in using what we learn about astrocytes to then say, can we come up with approaches in the future that would allow us to kind of dampen down those inhibitors of remodeling after an injury and allow the circuit to repair itself?
Yeah. I think it's fascinating. And I don't know, I constantly wonder about how people who want to hack themselves to live forever and make their brains healthy forever, I don't know. I'll stop there. But that's science fiction. We're working on things. But for the study that you published, it was called Astrocyte CCN1 Stabilizes Neural Circuits in the Adult Brain. And it gets at this question of stability versus flexibility, right? And so at a certain point, yes, you want to remember how to ride a bike, but maybe you want to learn how to ride a tricycle, or, maybe you want to ride a unicycle, right? So can you be flexible in the way that you learn something new? Can we learn new aspects of language? Or if we do, how does that happen? And so how can we maintain everything stably without forgetting what we had before? Can you talk a little bit about how you got to this study and what you did in it?
Yeah, so as I said, we got to it because these prior work had suggested or really shown that things outside the neurons could be kind of locking in plasticity in the adult brain. And things that were relevant to us was there was a study showing that if you inject an enzyme into the adult brain, an enzyme that kind of chews up all the proteins that live outside of the neurons, then just doing that was actually sufficient to allow more plasticity to be present. So it's kind of like a really key early study.
That's wild on its own. That's wild.
Yes. And there are other work showing that if you take young cells, so young type of neuron. They're called inhibitory interneurons, or young astrocytes, which we were interested in, and put those into the adult brain. Those are also able to increase the level of plasticity that's present. So kind of all these studies together were telling us, yes, there's something going on in the adult brain that's actively repressing plasticity, and maybe astrocytes could be one of those things. So that was kind of the hook to get us in there. And then the way we did the study was to kind of think about what do we know about the visual system. So everything we did was in the visual system, because there's been lots of work by neuroscientists here. So we know a lot about how it develops, when it has these periods of high plasticity called critical periods, and at what stage things get locked in. So what we were able to do was kind of had some predictions that if we thought astrocytes are cells that lock in this plasticity, then if they were actively doing this, then they would start making something at a higher level in the adult brain when you have this locking in. They would also downregulate it in conditions where you have remodeling happening. And in the visual system, if you kind of maintain in constant darkness the whole time, called dark rearing, this is something that actually will delay the maturation of the circuit and keep it more plastic. Or you can patch one eye, close it for a number of days, and then that allows the open eye to take over kind of the activity and remodel connections. And again, this is something that happens during this peak period of plasticity. So we just thought, well, if we profile astrocyte state across all these different conditions, when plasticity is high and plasticity is low, we should be able to identify things that change in the way we would predict. So that's what we did. So we used a technique that lets you measure gene expression, so RNA in specific cell types.
Cool.
So we could look at astrocytes specifically in the visual cortex. And what we did was we could look at each of these comparisons, and you saw many, many changes. But because we had 3 different comparisons where we kind of predicted things would go in the same direction, we could overlap them. And when we did that, we had very, very few candidates left. So I think coming in with this intersectional approach got us to a much shorter list of candidates.
So you got a list of candidates that were involved, and did you use AI in this to find these things? Were you just looking? Was it wonderful grad student labor?
A wonderful postdoc. And this was a while ago, so before AI was super hot. So we just looked things up on PubMed, looked things up on like UniProt, which is a protein database that's very well maintained and lists kind of all properties of things, various like resources that exist. And there really weren't that many candidates. There were 2 or 3. And Laura, who's the person who led the study, she then went in and validated them by asking if we found in this screen of where you're measuring like 20,000 genes simultaneously, if she then goes in and looks at each of them individually, which one actually replicates, which is important.
How do you see anything out of 20,000 genes when you're looking at microscopic brain cells? Like, that sounds incredible.
Yeah, microscopes.
I know, but that's amazing. It's like I'm honestly like the technology, the way it's advanced and what we're able to do now it's incredible. So you're able to label and be able to see all these different genes, different, was it genes or protein expressed from the genes?
Yep. So we looked at the gene level because that's what we had analyzed when we looked at all of them together. And we just picked 3 or 4 of them to go in and validate. So we weren't doing all of them together. You can now with the technology, but we didn't do that at the time. We just looked at a few. And this one CCN1 that we followed up on just had the expression pattern that fit what we were interested in. And also from the literature, even though it hasn't been studied much in the brain, it has been studied in other systems in the body. So there's quite a lot of research on it telling us what it can normally do, what it can function it can have. So there were lots of clues this would be interesting.
Yeah, so I've seen it pop up in a lot of different studies. Some of them related to cancer, some of them, just a multitude of studies. But the CCN1 is more of a structural protein. Is that right, or is it like a microtubule connector kind of protein?
Right. So it's actually a secreted protein. So then that's
Not
another reason we were interested in it, because I mentioned earlier all
extracellular,
the development.
not intracellular, extracellular?
Extracellular, sorry. Yeah.
Oh, okay. So between cells.
Between cells.
Hmm.
So it's like the things I told you earlier that in the young
Yeah.
brain are telling the neurons to make connections. This is something that in the adult brain the astrocytes releasing to communicate to other cell types. Yeah.
So neurons will release neurotransmitters across the synapse. Astrocytes, are they releasing similar kind of neurotransmitter chemicals, or is it this kind of structural signal, like like I'm making a real connection with you, kind of a thing?
So they can do both, actually. So there's quite a lot of literature looking at calling them gliotransmitters, because it's the transmitters but coming from glial cells.
Yeah.
And showing that it's not going to be like the same as like that fast communication between the neuron, but they can release substances such as ATP, which is actually a neurotransmitter itself, but can also be changed into adenosine, which is also a neurotransmitter, and that acts
Works
on the
great
neurons.
in the kidneys. No.
And that acts on the neurons then to change the likelihood that when that neuron receives an action potential, it will release a vesicle. So it's something we call release probability, and that's what it's acting on, and changing how effective that neuron is at communicating.
That's amazing. So basically you've got these scaffold structures or spider web, like Spider-Man spider webs that are coming from the astrocytes, that like little signals are traveling down. I'm going to send my paint cans or whatever. And they're there at the neuron to say, if they're there, then the neuron's going to be more excitable or less excitable. Like if inhibitory interneurons, we're talking about, they're less excitable. But I love that. So it's not the fat. That's part of the explanation. So about why it's not fast is because it is more of a physical transmission, or.
So the adenosine is pretty quick. That's more on the time scale of the neurotransmission, but it's not kind of this pre to post synapse type thing. It's signaling to the pre-synapse of the neuron, the one releasing the neurotransmitter, and changing the likelihood of it doing that. So it's kind of interfering or modulating that connection.
Yeah, a modulator. All right. So you've discovered, and other researchers have worked up this mountain of evidence to give you this idea to move forward. And you've looked at a bunch of candidates, and your postdoc, who is amazing, has looked through these. And Laura Sancho, was that her name?
Yep.
Yes. And so you found CCN1 as the target. So what did you do, and how did you really use CCN1 to go, this is it? This is the stabilizer.
Yeah, so we had kind of two main predictions or first experiments to do. And that was essentially to say, if this is something in the adult brain that actively stabilizes connections and stops remodeling, if we increase the level in the young brain when the circuit's plastic, this should be sufficient to repress remodeling. And that's exactly what we saw. Then we did the opposite experiment and same kind of hypothesis, and said, okay, if we let the circuit develop normally and then remove this factor from astrocytes,
how does it change?
If it's normally repressive, just removing it should now allow the neurons to undergo more remodeling. And that's also what we saw. So we kind of fulfilled our two criteria. This is something that was both necessary and sufficient to maintain this circuitry.
So when we think about the way that our brains change as adults, and we go into periods of change that we forget things. So dysregulation, Alzheimer's, dementia, etc. Is this the kind of signal that is going to be disrupted in those situations? Is this a hypothesis? Do we know this yet?
So I'd say for this particular signal, it's a hypothesis, but what we do know is we had a different study where we looked again at the gene expression profile of these astrocyte cells in the adult brain and the aging brain to ask whether maybe changes to these cells could contribute to things you just described in aging. And actually downregulation of CCN1 is something that happens in aging. So they're actually downregulating this stability cue, but we haven't done anything with that yet or followed up on it.
So this is a natural like downregulation with aging. It just happens as as that adult deterioration occurs in the brain normally. It's hard to, like, people don't want to recognize it's normal.
It is. Yeah.
But what are potential triggers that would make it occur sooner or more rapidly? And this is something you still need to look at.
Yeah, so what has also been shown by other groups is that this is something that gets upregulated after an injury such as a stroke. So it's kind of bidirectional. It seems like in cases where you kind of have inhibited this remodeling, that it's been upregulated. And in cases where we know there may be an instability of synapses, there's at least a correlation that it gets downregulated. As I say, we haven't like tested that yet to see if it's involved.
This is so fascinating. The regulation of it must be the most fascinating question. Like,
yes,
why?
Yes, and we don't know.
How are they?
That's the next project. It's like, why does it go up? Why does it go down? We don't know.
Right. So you're getting past, the old story, right? Not just support cells. These are essential to the way that our brain is structured, the maintenance of that structure, the plasticity of all of that. These cells are part of it. But what is this? What's the signaling? What is the conversation that's happening? Is there anything more than CCN1 that you're looking at? You said there were.
Oh, absolutely. Yeah. So in the case of, so you mentioned Alzheimer's. So in the case of Alzheimer's disease, there have been many studies recently looking at how astrocytes and other cell types change in patients with Alzheimer's disease. And some of the things that struck our interest, and we've also been looking at, is you probably talked about before or heard about, and you mentioned this inflammation. So the microglia as well, you get this inflammation that happens in neurodegenerative disorders. What struck our interest is that you also actually get a general downregulation of lots of things that normally maintain stable synapses. So besides CCN1, which we actually don't see changing, there are some other things that we've worked on in the lab. Another one of these glycogen proteins, this one's number 5, before we worked on 4 and 6.
Okay.
And this is one that's high in the adult brain and can actually help to maintain kind of synapse maturity and function. And it gets progressively downregulated as symptoms of Alzheimer's pathology progress. So we're interested in using this as a way to say, well, what if we stop it being downregulated? Can we actually maintain some of the functionality, maybe slow the progression of what's happening? So those are the sorts of approaches.
And so from the, very specificity of digging into this particular question to the more systemic aspect, are you talking with other neuroscientists about, that are looking into mitochondrial dysfunction? That's not your area, but it seems to be a big, possibility as one of the drivers of, the metabolism of cells leading to all sorts of dysfunction. And how would that tie into these therapies?
Yeah, so we haven't looked so much at mitochondria, but absolutely other changes that happen beyond the synapse. So astrocytes are really, really important for lipid metabolism in the brain. And they actually synthesize a lot of cholesterol in the brain. And you generally think of in the periphery, cholesterol is bad, but in the brain, cholesterol is really important and actually is really needed for the neurons to function and communicate. And it turns out within the brain, astrocytes and other glial cells are the main cell types that make it, and then they give it to the neurons for the neurons to function properly. And there's lots of evidence now and studies that people have shown that if we look in, for example, Alzheimer's disease, one of the major risk factors is this gene ApoE4, I'm probably familiar with. And one of the main roles of ApoE4 is to actually package cholesterol and take it, or ApoE in general, is to package cholesterol and give it to the neuron and other cells.
Huh.
And the studies now showing when you have the ApoE4 versus...
Here's your cholesterol.
Yeah, exactly. Versus ApoE.
That's great.
So ApoE4 is not as good at it as the other ApoEs that are kind of less predictive of having Alzheimer's disease. So that's kind of one way that this can be involved, and just other kind of lipid processing or other metabolic functions of astrocytes, which even though we kind of said, oh, we're interested in things like how to regulate synapses, all these metabolic functions are incredibly important as well for maintaining healthy brain function. And they also change in disease settings.
And the immune system, and like you mentioned, inflammation, there's like a huge feedback loop between the metabolism, the way that the lymph, the way that, all of everything in the brain, and it functions to maintain a healthy brain. What do you recommend as a neuroscientist studying?
Not a medical doctor.
Not a medical doctor, exactly. Yeah. Do I need to start taking probiotics? Like, I'm kidding. I'm not putting you on the spot here. That was terrible. That was a terrible joke. I'm putting you on a side conversation. So from the CCN molecule to these other possible proteins, other signaling, you're going to be looking at this stuff next. Where do you hope? Like, what's your hope for your research?
Yeah, the hope is that now that we and others have, I think, amassed this real kind of baseline knowledge of how astrocytes signal to neurons throughout the lifetime, but also in different brain regions, we didn't even talk about that, then now we have the basic understanding, right, of how this works. Yeah. So can we now use this in a way to target in different neurological disorders, to change synaptic state in the way that it needs to be changed, essentially to rescue the phenotypes that are seen? But we couldn't do that until we knew how they did this and what these signals were. So I think now we're getting to that point where we can start trying to apply these things and actually start thinking about things like Alzheimer's. How could we try and slow down some of the progression, for example?
You did just mention different brain regions. And so I'm just going to ask, as we get it, like with genes, with protein expression, we know that there are differences in different areas of the brain, that there are certain areas that express L-DOPA more than others, and so do you see a differential expression of CCN1 or these other factors in different areas, depending on how the areas are involved in what functions?
Yeah, so just on kind of on a high level, there's lots of studies now where people have looked at the gene expression of astrocytes across different brain regions, and it can be incredibly different. So the astrocytes, they'll still have all the baseline we're an astrocyte stuff going on, but they could have thousands of genes that statistically are different expression depending what brain region you look at. And some of that's intrinsic, it's because they arose in that brain region, they're programmed, but some of it is actually signals from the neurons and the environment. So that's like a whole interesting area of how these different cell types develop and specify.
Right, but in terms of being intrinsic, it's also different because it's doing different stuff than another brain region. So it's this brain region is functionally distinct, and the astrocytes are functionally distinct. So if you took astrocytes from a vocal production region and put them in the hippocampus, they wouldn't work the same way.
Right.
They might not work at all.
But people have done that, and that's what tells you some of it is programmed in the cell, and then they do change slightly on their environment. So some of it is coming from the signals that they receive. So that's why I say it's a combination.
Yeah.
Rather than
I think that's fascinating. So it's inherent. The astrocytes are like, I'm an astrocyte. I belong in a brain. I have a job. I could change it, but I like this job. I'm vaguely right there. So with this study and where you're going to be working in the future, what are your next steps? Where are you, like,
Yeah, I mean, next steps are one thing we were struck by in the paper is that when we permanently remove this signal from astrocytes in the adult brain, essentially the connectivity of the neurons in the visual system really regressed back to an earlier developmental stage. And that was actually bad because then the animals couldn't discriminate properly with a visual task. So that kind of was a lesson to us, and I'm sure you see in many other studies that just permanently getting rid of something isn't the right way to go if we're trying to think about eventual ways that we could use these things therapeutically. So we're interested in the lab in trying to develop techniques where we could go in and kind of in a transient way remove these inhibitory proteins from outside the neurons, allow them to remodel, perhaps by also combining with like a new learning task to get the appropriate connectivity, then allowing the neurons to lock back in again, essentially. So trying to think of tool development, ways we could transiently disrupt these inhibitors.
I think that's going to be so important for the therapeutic use of these future treatments, to be able to actually lead to something useful. Like we've had instances where we've done cell transplants where we're trying to put neurons in the spinal cord, where we're trying, but then it causes problems. We haven't understood the rest of the picture. So I actually really appreciate the fact that you mentioned like creating the right cell type, the right environment, and also a visual training task so that however it works out, it's a holistic situation, organismal, as opposed
it's not going to be one thing.
Not just in the brain.
So,
yeah. How do you look at that slicing and dicing of organism versus brain versus behavior? Where do you draw the line on how you are looking at your questions? Are you more molecular, or do you derive from where you are as a person?
I think we're cell molecular neuroscientists, but we appreciate you have to understand the outcomes. So you need to look at the circuit level and the behavior, and more now we and others, you have to think about the whole body. So think about body state, think about inflammation in the body, how, because the brain senses it, the state of the cells will change. So really it is, even though our core is on the cell molecular, we don't limit ourselves to that. We just do kind of what we consider the right experiment for what we're trying to look at.
You've done some important work though, so I really appreciate that. Your lab's done some really great stuff. Is there anything else that you want to tell us about your work or your lab?
I have an amazing lab.
Obviously. Oh my gosh.
The people are amazing.
You've got an incredible lab with with amazing grad students, postdocs, people that you're working with. You're asking incredible questions and using technology in all the best ways. Yeah. How do you want people to think about your work? What's a take-home message?
I think the take-home message is if you want to repair the brain, improve function in different neurological disorders, or just improve in healthy aging, then if we consider all the cell types in the brain, including astrocytes and how they regulate the neurons, this gives us new targets that we can develop therapeutics around to eventually have impact on patient health.
I think that sounds like it's a huge benefit. Having more options is always better.
Yeah, exactly.
Thank you so much for spending this time with me. I really appreciate getting to talk with you about your work. And as somebody who has a background in neuroscience, I have to say, I really enjoyed this conversation and learning some new things. So thank you.
Thank you.
And where can people find you if they want to learn more about your work?
At the Salk Institute in La Jolla. So I'm on the website.
Yeah, Yes, you are on the website, and you have a lovely lab website that people can find all sorts of information about your work and the people who work there. I hope that people do decide to check it out and learn more about astrocytes, which I think are an interesting, fascinating thing. Thank you so much again, and I hope you have a wonderful night.
You do.
Enjoy. Be safe getting home.
Bye.
Good night. Bye. Hey, everybody. This is This Week in Science. Thank you so much for joining us for this incredible interview, learning more about the brain, stability, plasticity, and the components that are involved in deciding how that all works. So glad to have you here. Thank you for joining me. If you are enjoying the show, please head over to twist.org and help support us in an ongoing way by clicking on that Patreon link. Alternatively, we have our Zazzle store. There's a PayPal link as well. Twist.org is where you can find ways to help support the show in an ongoing fashion. Really can't do this without you. Thank you for your support. Thanks, everyone. Normally, I would bring Blair to the stage now for Blair's Animal Corner, but unfortunately, she is out for the evening, and so it's up to you and I to talk about science. I have a few stories before I head home tonight, so if you will allow me and give me more of your time. Here we go. The NSF-DOA Vera C. Rubin Observatory has launched its real-time discovery machine, it says, for monitoring the night sky. As of last night, the 24th of February, the Vera Rubin Observatory had released what it calls its first alerts. These are alerts documenting changes in events spotted by the observatory. So it's taking picture of the southern hemisphere. So many pictures. And if there are differences between the pictures, then it alerts people and researchers, observatory groups, all sorts of people can sign up to be alerted about changes that they witness. So they're going to be looking for things like supernovas, variable stars, galaxies, dynamic galaxies, all sorts of things. And every night we'll be taking something like 10 terabytes of picture data, scanning the skies every few seconds. And within 2 minutes, it will have compared the images with the images previously, determined what is different, if it is different, and if somebody has an alert or if there's a need to alert, then it will alert the person. So where this is going that is really, really cool is that it is going to speed up how astronomers, how scientists who are interested in looking for things like supernovas, it's going to speed up how they're alerted to this information. And so there are going to be these pings, and Rubin will send them out. And almost immediately, within 2 minutes of going through their algorithmic stack, researchers will be alerted to the changes that are taking place. So the Vera Rubin Observatory is going to be looking at stuff moving forward. Last night, 24th of February, it issued 800,000 alerts. And so this is the first time it's done this. It's going to be ramping up to probably something like 2 million alerts every night. The whole stack is thanks to an amazing collaboration between different groups, the data brokers that they've involved for Rubin. There are 1, 2, 3, 4, 5, 6, 7, 8, 9 data brokers. These brokers are the ones that are approved by the community and are working for the community. You can access alerts through the brokers. There are also downstream services. They're an international network enabling this real-time exploration, near real-time exploration of the sky. There are also collaborations with Zooniverse and others that are going to be allowing more community interaction. However, they've definitely got some big intelligent software programs working to make all of this happen. Yeah, turn off your phone, Fada. You don't want 2 million alerts a night. Exactly. But this is very exciting because we haven't looked at the southern hemisphere as much as we should, or the night sky of the southern hemisphere as much. And additionally, this is going to be, like I said, super fast. It's going to speed up how scientists, researchers, anybody really sees things, is aware of things in the night sky, and is able then to follow up. So if you are aware of something, immediately you can collaborate and make sure that you can move forward on any planned research that you want to do moving forward, which that's kind of exciting, speeding that up. In terms of speeding up, NASA is slowing down. So they were wanting to launch Artemis today. However, the Artemis 2 rocket was rolled back to, it's being rolled back to the hangar for repairs, and the Artemis 2 mission to the moon, manned mission to the moon, personed mission to the moon is not going to happen in March. It's just not going to happen. So they were looking to launch as soon as March 6th. However, that will probably not happen, and they are starting to look at April. Dun, dun, dun. Most likely. We'll see how that goes. And then I have a quick study related to my name, because of course, every time somebody does research related to the Bouba Kiki effect, I have to tell people about it. So researchers decided to look at what is known as the Bouba Kiki effect. If you haven't heard of it previously, people and some other species tend to respond to the roundness of the booba sound. So if presented with a round versus spiky image, while the word booba is being said, individuals tend to respond to booba for round more often. Kiki tends more spiky. So this new study, they wanted to look at not mammals, but look at a different taxa. So birds is where they came in. They looked at baby chicks and wanted to see how baby chicks respond to the sound of booba or kiki. And they determined, according to the study, that I'm going to see if I can get into these wonderful videos that they have to be able to share. Here, if you can hear this or not. So they have a funnel-like shape that they put the little chick into, and then a night light that shows a spiky shape and a roundy shape.
Kiki.
And they say my name a lot.
Kiki. Kiki.
And then they look to see
Kiki.
where the little chick
Kiki.
decides to go. And
Kiki.
according to their
Kiki.
study,
Kiki.
there's a
Kiki.
difference.
Kiki.
Yeah, you hear this? It keeps going. Kiki, Kiki, Kiki. There's a lot there. They have many videos for all of the chicks that they tested, and their methodology. They used classical music as the control, as opposed to actually using these words of Kiki and Bouba for the chicks. Classical music. The chicks appeared to like the roundy shape of the booba much more than the spiky shape of the kiki. The chicks just really like the roundy shape more than the spiky shape. And I'm not convinced that they took that into account enough for their assessment, but they were published in Science. So, I mean, that's something. Um, but the study published in Science says that even chicks, even the bird brain, responds to roundies for Bouba and spikies for Kiki. But um, there was a preference for the Bouba. I don't know. Is it the Bouba or is it the round shape? I'm still not convinced. However, what else did I want to talk about from chickens and the Bouba Kiki? Oh yeah, two cool brain studies that came up this week. Researchers looked to determine how becoming a pro at something, becoming good at something as an adult, and this kind of ties in with the interview from earlier, how neuroplasticity is involved in uh, expertise. And they looked at birders. And so in this study, they did an fMRI, functional magnetic imaging study, showing birder experts bird pictures, novices got the bird pictures. They even showed the birder experts local birds versus not local birds to see how the brain changed in the way that it was trying to assess the information. And they determined in this very not large study, 29 individuals, 15 female, ages 24 to 75, wide age range, uh, adult for sure. They matched them to novices and they looked at this diffusion weighted MRI information and determined that birding has the same kind of brain impact that other expertise based activities also have. So it's really the areas of the brain. There's cortical in the cortices, the areas of the brain that are involved in attention and understanding and and your self-control, that the structural remodeling, functional tuning, how processing happens, it is tuned, it is winnowed, that there is plasticity that occurs when you work really hard to become an expert at something. And so they found differences in different brain regions in the experts versus novices. And so they suggest that convergent structural remodeling and functional tuning in service of expert performance occurs across the lifespan. So not just when you're young, but as you get older, and it occurs across your age. So this is, I think, really, really interesting as we come into thinking about the interview and that information that the astrocytes might be involved in this modeling and fine tuning as long as we're alive and the neurons are there. It just helps the neurons figure out what to pay attention to and when. And of course, this is me, coming up with a way to explain it. But I think it's really an interesting and important thing to get excited about, that your brain isn't just gonna fall apart as we get older, that we're not just born with all the cells that we have and then they start dying, that we can actually change as we get older. All these things are important. And my last study for the night, this is 1 by land, 2 if by sea. I see you. Yes, you can tell if something is a bird 4 out of 5 times. I think you have some great expertise. Very well done. Last study of the night is related to monkeys drinking alcohol. So this study is really interesting because we've talked before on the show about chimpanzees and other primates ingesting fermented fruit. And there was a study a while ago that suggested that chimpanzees probably imbibe or eat probably the equivalent of a beer or a glass of wine a day. And so the researchers who did this original study really wanted to find out, what's the alcohol content that's coming out in the urine of the chimpanzees? So a graduate student named Alexi Morrow slept in the trees, was out in the field, had to go out and stand underneath the primates in the forest and collect their urine. He collected it off of leaves. He held out a cup on a stick, collected it off of that. He got it out of little puddles on the ground. If any job is an interesting grad student job, this is it. Being in Uganda collecting primate urine. The researchers actually found they used a simple test that is looking at ethyl glucuronide, which is a byproduct of alcohol that we use very often for humans. Immunoassay test strips very simply measure alcohol levels for us. They use the same strips. And they were able to find this byproduct above the 500 nanogram per milliliter threshold. And this is about 1 to 2 drinks for humans. And so they actually were able to confirm that chimpanzees are out there eating enough fermented fruit to have a blood alcohol level that is 1 to 2 drinks in. So they've got evidence of not just watching chimpanzees eat fermented fruit. They have now assessed their urine, and if there's alcohol in the environment, then the chimpanzees may be experiencing it. We don't know if it is exactly analogous to humans because of the way that they ingest it. It is fermented fruit, and so they tested a bunch of these apples that the chimpanzees seemed to like, but they didn't have, and even the ones that were on the ground didn't seem to have enough of the sugar alcohols in them to really be the reason, unless the chimpanzees are eating a massive, massive amount of these fruits. So the question is, how do you get this urine concentrated with alcohol if you're not eating something that is very, very fermented and has alcohol concentrated in it? You have to be eating very quickly. You have to, there's a whole bunch of factors that go into it. But there are drunken monkeys. It's true. I'm not saying monkeys, apes. The chimpanzees, they might be drunk. 1 to 2, depending on how they handle their alcohol. That's very interesting. The really interesting aspect to this, though, is that they did not find urine in pregnant or female monkeys that were in estrus. So those that were ready to have babies, and juveniles. So young chimpanzees were not ingesting the fruits that had the fermented alcohols in them, and neither were female chimpanzees that were ready to procreate. So this is, I think, very interesting. Why does this all happen? Is it because they're not eating those fruits? Are the male chimpanzees bogarting the fermented apples? We don't know the answer to this yet. So I think that the graduate students need to spend more time in the forest. And that is me for the day. Do you have comments, questions? Let me know. Thank you for being here tonight. I appreciate the fact that you have joined me for the evening. I hope you had a wonderful evening. I hope you had a wonderful week. Thank you for being here. Shout outs to Fada for doing social media and show notes, Gord, Arnlore, others for making sure the chat room is a great place to be. All of you in the chat, thank you for chatting and all your comments during the show. Identity 4, thank you for recording the show. And Rachel, thank you for editing. I also have to thank our Patreon sponsors for all of their help ensuring that this show keeps going. Thank you for making sure that we're able to keep making it go. Aaron Anathema, Adam Mishkan, Alan Viola, Alan Swanson, Alan Viola, Ali Coffin, Andrew Swanson, Arthur Kepler, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Daryl Myshax, Dave Wilkinson, David Youngblood, Don Mundis, Donovan Styles, aka Don Stylo, E.O. Ing Mundell, Eric Knapp Flying Out, Fredis 104, G. Britton Lattimore, George Chorus, Greg Briggs, Jack Jason Olds, John Atwood, John McKee, John Ratnaswamy, John Gridley, Ken Hayes, Kent Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessen-Flo, Mark Jenkins, Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Philip Shane, Pierre Velazar, Richard Badge, Robert Norland, Robert Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean, Clarence Lamb, Shu Brew, Stephen Albrown, Steve Leafsman, aka Zima, Sue Doster, Teresa Smith, Tony Steele, and Vegard Jeffstad. Thank you for all your support on Patreon. Can't do it without you. If you're interested in helping us out on Patreon, head over to TWIS.org and click on that Patreon link. We will be back next week. Hopefully Blair will be back too. We'll be broadcasting live Wednesday, 8 p.m. Pacific time on YouTube, Facebook, and Twitch. And if you want to listen to us at a podcast, make sure you head over to your favorite podcast platform and look for This Week in Science and get your friends to subscribe too. If you want any more information about the show and anything you heard, make sure you go to twist.org. You can sign up for our newsletter that will probably come someday. And we love your feedback. Let us know if there's something you want us to cover or address or an interview. And you can send us an email at our email accounts. Just make sure you put TWIS in the subject line so the email doesn't get spam filtered into one of those, non-functional presynaptic pathways where it doesn't really tell neurons to do anything and it just sits there. Yeah, so maybe not do that one. Yeah, make sure you put TWIS in the subject line. Love your feedback. And I look forward to discussing science with you again next week. If you learned anything in the show, please remember, it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science. This Week